Hey guys, so this is the Enzymes lecture video. If you haven't opened up the student note page already, go ahead and do so. Hopefully the sub has some hard copies for you, but we'll go ahead and get started. So before we get into what enzymes actually are, I want to talk about what a catalyst means. So a catalyst is a substance that speeds up the rate of a chemical reaction without actually entering the reaction itself. Okay, so catalyst can be in two forms. It can either be inorganic as heat or in an organic way known as enzymes. And most catalysts that we're going to talk about, and actually that are in this world, are actually organic. So I want you guys to go ahead and pause the video, and I want you guys to watch the Amoeba Sisters enzyme video. And just go ahead and click on the link and you should be able, well, actually, never mind. This is a lecture video. So let me go ahead and play it for you. Okay, so before we actually get into what enzymes are, I want you guys to watch the video and just kind of get an idea of what we're going to be talking about.
Odd to have a favorite protein? Well, I don't think so. Probably because my favorite protein happens to remind me of one of my favorite childhood games, Pac-Man. If you haven't played Pac-Man before, then chances are we're much older than you. But now you can play it on Google. Just Google Pac-Man. It's a Google Doodle. Anyway, I digress. But see, in Pac-Man, you have this little character. It goes around, finds these pebbles, and the pebbles fit right into it. Well, a lot of illustrations that you will find of enzymes, they happen to look, at least to us, a lot like Pac-Man. I remember P for Pac-Man and P for protein. Most enzymes are proteins. Now, in the game, we mentioned there are these little pebbles that Pac-Man goes after. Well, enzymes have a specifically shaped area called an active site where items can bind. The items are called substrates. It's very specific binding because the active site is specifically shaped for the substrate that binds there. Very specific. So, what happens when a substrate binds an enzyme? Well, usually the substrate is held there with some weak bonds because it's not going to stay there forever. And something called induced fit will happen, which means the active site can change its shape even more to bind that substrate, like an enzyme substrate hug. The enzyme can either build up or break down the substrates that specifically bind to it, and we call the resulting item the product. An enzyme has the ability to really speed up reactions that technically could happen on their own, but with the help of enzymes, they can be sped up to make processes effective for life. Let me give you a great real life example. The enzyme lactase. Another really cool thing about enzymes is that they often end in ASE, like lactase. Many sugars, on the other hand, end in OSE, and lactose is an example of a sugar. Lactose is a disaccharide, meaning it contains two sugar molecules bound together. We don't actually digest it so well in that form. It's big. The enzyme lactase has the ability to break lactose down into smaller parts that our body can digest. And this is a lot better option than waiting for a chemical reaction with lactose to happen spontaneously. And with the lactase enzyme, lactose can be broken down quickly and digested. Now, there are some people that do not produce enough lactase. They can be what we call lactose intolerant, which means that if they consume foods that have lactose, such as milk, it can make them sick. They can't break the lactose down efficiently without lactase. In that example, one thing to point out, lactase, the enzyme, can break down a lot of lactose, the substrate. The lactase doesn't get used up in the reaction. It's still there. We call enzymes a catalyst because they can be used over and over and over and over in the reaction. By the way, your digestive system uses all kinds of enzymes. You have lipase, and that breaks down lipids, which are fats. You have amylase, which breaks down starch. You have protease, which breaks down proteins. So, as you can see, the digestive system is very involved with enzymes. Another thing to point out is that enzymes don't always work alone. Sometimes they get some help. some often underappreciated but essential little helpers are called cofactors and coenzymes. They may bind to the substrate or to the active site. They help the enzyme do its job of building up or breaking down substrates into products. Now, you didn't forget our Pac Man analogy yet, right? In the game Pac Man, there are these ghosts. And when they touch the Pac Man, it makes this sound. It's like, no, 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 no. And the Pac Man shape gets all distorted in the process. So, what does this have to do with enzymes? No, there aren't ghosts around, but enzymes do have certain ideal conditions that they like. For example, an enzyme that is in your stomach would have an ideal pH that is very acidic because the environment in your stomach is very acidic. Different enzymes have different ideal pH and temperature ranges. If an environment changes out of an enzyme's ideal pH or temperature range, then something that reminds me a lot of that horrible sound I tried to make can happen. The enzyme becomes denatured. That means its shape becomes distorted. It can no longer bind to its substrate. It can no longer work correctly. It is finished. Well, that's a dramatic end to enzymes. Keep in mind that if you have an interest in this topic, many medical researchers have a large focus on enzymes. Enzymes regulate a lot of the body processes, and many diseases can involve specific enzyme production or the lack of it. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Alrighty. All right, now let's go ahead and get into enzymes. So pretty much I'm just going to kind of go over what she was explaining. So enzymes is an organic catalyst made of protein. Now remember that catalysts are a substance that speeds up the rate of a chemical reaction without entering the reaction itself. So enzymes, those are organic catalysts, and enzymes are proteins. Okay, I know we learned about macromolecules last class. We talked about carbohydrates, we talked about fats, lipids, proteins, and nucleic acid. So enzymes fall under the category protein. Now, like she said in the video, most enzymes do end in ASE. So whereas like carbohydrates, they end in OSE, like glucose, sucrose, fructose, cellulose. Enzymes end in ASE, and we're going to go over some examples here in a little bit. And what enzymes do is they work by lowering the activation energy of a chemical reaction. So what this means is it lowers the amount of energy needed to start the reaction so the reaction can happen a lot faster. Okay, what does all that mean? So you'll notice in this picture that we have a really tall hill, right? So this red line acts as this activation energy, okay? Now let's pretend I want to push a ball. If you're following my marker here, let's pretend I want to push a ball up this hill, right? There is a lot of energy that needs to go into that, right? A lot of muscle, a lot of strength, a lot of energy to try and get that ball up the hill and then all the way down the hill, right? So what enzymes do is they help lower this activation energy so that the ball can happen or can go over the hill a lot faster. So in other words, enzymes help lower that activation energy so that the chemical reaction can happen a lot faster. Okay. So how do enzymes work? So enzymes act on molecules called substrates. Okay, so in this picture here, this yellow thing is an enzyme, and then we have this green thing over here, which is called a substrate. And a substrate and an enzyme need each other in order for a chemical reaction to start. And what substrates do is they bind to their substrate at the active site. Okay, now every enzyme will have what's called an active site. And typically enzymes are larger than their substrates, like you can see here in this picture. Enzymes fit into their substrates like a key fits into a lock. It is very specific. So you see how like these are a perfect match, right? Like this substrate, the shape of it will fit perfectly into this active site on the enzyme. That's like the perfect ideal situation. This is called the lock and key model of enzyme action. So remember in the video how she was talking about Pac-Man and how Pac-Man has like the open mouth and then the substrate has the perfect fit to get into that open space. That's kind of like the same idea of a substrate and an enzyme. Okay, so steps of enzyme reaction. Guys, you do need to know this for the test. I for sure know this is on the test. So the first step is an enzyme joins with its substrate. That's the very first step. So you can just kind of follow along with the pictures here. This is step one. Enzyme joins with its substrate. Step two, enzyme and substrate form what's called an enzyme substrate complex. This means that they are attached, they're a perfect fit, we're ready to go. Step three, enzyme action takes place. So you'll notice that the colors are totally different in the enzyme products complex. And notice how I said products, you guys. So here we start out, your substrates will start out as what's called reactants. And at the end, they will be called products. Okay, we'll get into that in a little bit too. But this is like your chemical reaction taking place. And then the fourth step is the enzyme and products will separate. The enzyme is unchanged and can be reused. Okay. So notice how the products, or I'm sorry, let me kind of backtrack. The substrates, right, turned into products and they are totally different now. And now they're attached from the enzyme. They're ready to do their job. But you'll notice that the enzyme is unchanged and it can be used again. So we'll go find another substrate that fits perfectly into its active site and then do another chemical reaction, okay? So 4 steps that you need to know on how enzymes are in action. Okay, enzymes are so picky. They only work under specific conditions. So I kind of actually like this picture over here. It says, I'll take a latte medium, no bigger than that. And the cashier's like, hot, or no, I'm sorry. I'm so sorry. This is the enzyme. So hot, but not too hot. And with extra foam, not that much. Just a smidge of cinnamon, more than that, too much. Do it again. I'll denature if it's not perfect. How many of you have a Starbucks order kind of like this? And it's okay if you do. That means you're exactly like an enzyme, meaning that you are very particular with what you want and your conditions. So, like I said, enzymes are super picky, and there are certain factors which affect enzyme action, and those are pH levels. We talked about that during properties of water. Temperature, and then concentration of enzymes and substrates. So if the pH levels and the temperature and the concentration are not specific for that enzyme, then the enzyme will denature, okay? Now remember, kind of going back, you guys, sorry. Remember, denature is like meaning to destroy. I think I'm going to talk about that here in just a bit. Each enzyme in your body or in any living thing has a specific pH in which it will work. Different enzymes work at different pH levels. So, for example, in your guys' stomach, you have an enzyme called pepsin, and it works best at a pH of 2. Okay, and the reason why is because of digestive juices that are in your stomach, and your stomach is very acidic. So remember how we talked about from 1 through 6, that's considered like the acidic levels on the pH scale, right? So if it has a pH level of 2, that means it's like it likes a very acidic environment, okay? Now, you also have an enzyme called amylase, which is an enzyme found in your mouth. And just kind of a little fun fact, you guys, if you just had breakfast, like let's just say you had a burrito, right? Well, think about it. Like that big bite that you took, do you think that could fit down your esophagus? To me, not really, right? Your esophagus is pretty like small, right? So you need something to help break it down and make it a little bit smaller. So believe it or not, like when you have the food in your mouth, your nervous system will send a trigger to your saliva and allows that amylase to be triggered. So amylase actually helps break down your food and helps it make it smaller so that it can fit into your stomach, into your esophagus and then into your stomach and so on and so forth. Also, a little fun fact, you guys, I don't know if I have, I don't know about you, but sometimes like when I lick my lips too much, I'll notice that they start to get dry. That's because I'm putting amylase all over my lips, and amylase, what it does is it breaks down, right? Enzymes break down things. So that's why your enzymes get so, I'm sorry, your lips get so cracked if you lick your lips too much. So just kind of to be cautious about that. But anyways, kind of getting back on track. Enzymes love to work at a pH level of 6.8. Okay, so they kind of like a high acidic, more neutral kind of area. So let me ask you this. If we were to put amylase in your stomach, what do you think would happen? Or what do you think if we put pepsin into your mouth? What would that do? Well, remember that enzymes are very picky, right? And one of the reasons was your pH levels. So if pepsin is in a pH level of 6.8, it's going to denature. Same thing with amylase. If amylase is not at a pH level of 6.8 and is in a pH level of 2, it's going to denature, aka be destroyed, and it can't be used ever again. Okay. All enzymes have an optimal temperature at which they work the most effectively. Okay, so just like pH levels, temperature, same thing. Okay, optimal temperatures for humans is 37 degrees Celsius, whereas optimal temperatures for dogs is about 101 to 102 Fahrenheit. So 37 degrees Celsius, you guys, that's like roughly 96 between 98 degrees Fahrenheit. So if you get sick, right? Or you can actually kind of see this. Heat energy causes more collisions between enzyme and substrate. Optimal temperature for humans is roughly around 37 degrees Celsius. Enzymes denature at high temperatures, so rate falls rapidly. So when you guys have a fever, any of you have had like the fever before, or like a flu, right? You get like a body temperature, like you're really, really hot. Okay. Well, if your body is starting to get hot, remember your enzymes like a certain temperature. So if your body's getting really hot, they're going to denature and they won't be able to work. Hence why you like if your enzymes aren't working, you're going to feel sick, right? So that's why you take like, medication and supplements and whatnot. Okay, so like I said, what happens if temperatures get too high? Enzymes change shape, kind of like melting almost. And when this happens, we say the enzyme is denatured. If the enzyme is not shaped correctly, it cannot bind with its substrate and can no longer do its job. So you can see here in this picture, right, we have an enzyme, we have the active site, we have the substrate. It looks like a really good, perfect fit. Uh-oh. But then we get a fever or the flu, and our enzymes start to denature, and look what happens to the active site on the enzyme. Do you think this substrate can actually fit on the active site on the enzyme? No, it can't. So therefore the chemical reaction can't occur. And when I talk about chemical reactions, you guys, that's like digesting your food, just certain like activities happening in your body that you are unable to do. So enzymes are really, really important when it comes to just your overall health. Okay, same thing here, you guys, with pH changes, right? We have a perfect case scenario, but then if you put an enzyme in a different pH, look what happens to the active site, therefore the substrate can't bind. Last but not least, concentration and enzyme. So as concentration of both the enzyme and the substrate increase, the rate of enzyme action increases to a certain point, and then it will just start to level off, okay? So again, if the concentration of it just keeps continuing to increase, then eventually it will just start to level off. It won't go down, it'll just start to level off, kind of like what's it called, the carrying capacity that we learned in the ecology lesson. It's pretty much like the same situation. So why does leveling off occur? So if the concentration of the enzyme increases, the reaction rate will initially increase as well. However, as all of the substrate is broken down, the excess enzyme has nothing to combine with, right? So the reaction rate will just level off. It won't decrease, it won't go away, it'll just level off, okay? If the concentration of the substrate increases, reaction rates will also initially increase as well. However, if there are not enough enzymes to act on the increasing substrate, then the reaction rate will level off as well. Okay, So here are some summary questions that maybe just kind of go ahead and pause my video, turn to your partner, and just maybe like quiz each other really quickly. Like, what type of biological molecule is an enzyme considered? Why is enzyme action similar to the action of key fitting into a lock? What is the difference between a substrate and an active site? Why are changes in pH and temperature critical to the proper functioning of an enzyme? So if you want to go ahead and just turn to your partner and just kind of like try and answer these questions without looking at your notes. And if you do need to look at your notes, not a big deal, but we can go over this when I'm in class with you next. All right, that's it. So I think you guys have a worksheet on Brightspace. Let me see if I can go ahead and pull that up for you. Okay, so biochemistry. Guys, just a reminder that I do have all these videos here posted for you. I do have enzymes down here. So if you want to watch that video again, you have the link there. Okay. All right, so you have macromolecule and enzymes review. And then you just have a couple vocab questions on the bottom there. Okay. So let's see, it looks like you have some matching here. It's asking you to watch another Amoeba Sisters video. I know, I'm sorry you guys aren't a big fan, but I like it. I think it's really, again, I like how she simplifies it. So thank you for being troopers. And you're gonna go ahead and just write C for carbohydrate, L for lipid, P for protein, N for nucleic acid, along with its corresponding definition. You have some fill in the blanks here, some labeling, and then just a couple questions down here. Okay, so this will be due at the end, at the beginning of next class. So I will see you guys on Thursday morning. And there should be a Shelby box ready to go for you, okay? All right, well, if you guys have any questions, please don't hesitate to send me an email. Otherwise, I will see you on Thursday. Just a heads up, we do have a lab next class, so please come prepared, ready to go. And then we do have your test, which is at the beginning of next week. So just a heads up that is coming up. But otherwise, I will see you guys next class. Thanks. Bye.